Greetings, dear learners. This is Dr. Steffi, Assistant Professor, PG and Research Department of Microbiology, Kaveri College for Women, Autonomous, Trichy. In this module, we will be discussing about the 3-domain and 6-kingdom classification in detail. The 3-domain system is also known as Karl-Voss classification system. The 3-domain of Karl-Voss classification system includes archaea, bacteria, eukaryote, and the 6-kingdom system of classification includes archaea, eubacteria, protista, fungi, plantae, and animalia. Now coming to this classification system divides the life based on the differences in the 16S ribosomal RNA structure and as well as the cell's membrane lipid structure and its sensitivity to antibiotics. Their main difference from earlier classification system is the splitting of archaea from bacteria. The evaluating rRNA structure is very helpful. As a result of rRNA molecules throughout the nature, it performs the identical function. Their structural modifications are very little over time. Subsequently, similarities and dissimilarities in rRNA nucleotide sequences are a very good indication of how it is associated or unrelated completely, which is different from cells and organisms. In this classification, Karl-Voss uses 16S ribosomal rRNA as a chronometer. This is because it is universally distributed by means it is present in all the species. It is functionally similar in all organisms. It can change its sequence slowly. Its sequence can be aligned or matched up between two organisms. Now coming to the pictorial representation of the 3-domain classification system. The pictorial representation includes bacteria, archaea and eukarya kingdom. Examples of bacteria include purple bacteria and flavobacteria. Examples of archaebacteria include methanococcus and thermococcus. Examples of eubacteria includes flagellates and trichomonads. Now let us discuss about the domain archaea. The archaea domain includes all prokaryotic cells. They lack nuclear membrane. They have distinct biochemistry. They contain rRNA molecules from bacterial cells. The archaea are considered as the oldest species of organism on Earth. They can survive in extreme harsh environments that differentiated them from other domains. The cell wall of archaea lacks peptidoglycan. Archaea also contains either leakage in their membranes. Now let us discuss about the domain bacteria. They are also prokaryotic cells with bacterial rRNA and contain diacylglycerol diester lipids in their membrane. They are also called as eubacteria or true bacteria. Their cell membranes contain ester linkage between unbranched fatty acid chains and glycerol. Their cell wall is made up of peptidoglycan. They are present in the form of 5 phyla, which includes proteobacteria, cyanobacteria, eubacteria, chlamydias and spirochetes. Now let us discuss about the domain eukarya. These are eukaryotic cells with a membrane-bound nucleus. Their membranes contain their membrane, which includes unbranched fatty acid chains and glycerol. They lack peptidoglycan on their cell wall. Eukarya inhibit the antibacterial antibiotics, but they are sensitive to antibiotics that affect eukaryotic cells. They are presently of 4 types, which includes protista, fungi, plantae and animalia. Now it's a time for recap. In this classification, Karl-Voss uses 16S ribosomal rRNA as a chronometer because it is universally distributed by means it's present in all the species, it is functionally similar in all the organisms, it can change its sequence slowly, and finally its sequence can be aligned or matched up between two organisms. There are three domains of life domains. Includes bacteria corresponding to domain Eubacteria, domain Archaea corresponding to kingdom Archaebacteria, and domain Eukarya corresponding to kingdom Fungi, Plantae, Animalia, and kingdom Protista. Dear learners, thank you for your patience listening. We will meet up soon in the next module.